Today, we will be discussing a recently published paper on Alzheimer's disease. Alzheimer's is the most common form of dementia and is nearly always caused by aging. There is no current cure for the disease, but lab groups are constantly studying the mechanisms of this disease to potentially find a therapeutic target or cure. One hypothesis regarding the onset of Alzheimer's is known as the Tau or TAU hypothesis, and it suggests that Alzheimer's is caused by the breakdown of the neural cytoskeleton. Specifically, Tau is a protein that stabilizes microtubules of the cytoskeleton, and this hypothesis suggests that overphosphorylated Tau protein forms aggregation inclusion bodies and prevents Tau from stabilizing the microtubules, leading to degradation of the cytoskeleton, and thus Alzheimer's onset. The purpose of this article is to reveal specific changes in human DNA that could potentially result in higher rates of Tau aggregation, and thus Alzheimer's onset. Specifically, these researchers discovered by a genome-wide association study a variant of the gene coding for cyclin-dependent kinase 5, regulatory subunit associated protein, or CDK5-RAP, called RS10984186, that results in overexpression of this gene. This protein is known to activate cyclin-dependent kinase 5, which is responsible for phosphorylation of the Tau protein. When Tau is overphosphorylated, it can aggregate. Thus, when CDK5-RAP is overexpressed, this can result in the buildup of this aggregation, or NFT, neurofibrillary tangle. We will focus on Figure 4. In Figure 4, the researchers investigated the abundance of CDK5-RAP mRNA in comparison to the density of NFT in the frontal cortex. They plotted the data comparing these two variables for several individuals. The slope of the graph depicting NFT density as a function of CDK5-RAP prevalence was determined to be 0.21, and this demonstrates that a positive correlation exists between these two variables, specifically that there may be some aspect of increased abundance of CDK5-RAP mRNA that could potentially cause buildup of NFT in the frontal cortex. These researchers and the scientific community as a whole had already known that NFT buildup is associated with memory loss in Alzheimer's patients. So, instead of simply correlating a certain allele with Alzheimer's prevalence, in this experiment the researchers were actually able to take the association a step further and correlate the CDK5-RAP mRNA abundance with a potential indicator of Alzheimer's. This still only shows correlation, not direct causation, but the specificity of the analysis does indicate a higher likelihood that there is some molecular relationship between CDK5 and Alzheimer's. The rest of this paper goes on to discuss the other significant results that indicate a positive correlation between the RS10984186 allele of CDK5-RAP and the risk for developing Alzheimer's. For example, Figure 1 shows a genome-wide association study indicating the probability that there is a correlation between the RS10984186 allele and the risk for developing Alzheimer's. Additionally, Figure 3 shows the high abundance of CDK5-RAP mRNA in various brain regions in Alzheimer's patients compared to control patients. Thus, the overall conclusion is that this polymorphism may potentially have a significant impact on the onset of Alzheimer's. The field of Alzheimer's research is very wide, and there are several hypotheses that people support in reference to the exact mechanism of the disease. The Tau hypothesis is only one, but this paper indicates that further investigation of such a hypothesis may be wise. Additionally, it might prove useful to further elucidate the molecular relationship between these macromolecules, as this pathway may, in the future, serve as a potential therapeutic target for Alzheimer's.